All right, so we are live. All right, so today's seminar speaker, I'm happy to introduce, is Bethany Rader. Bethany and I know way too much about each other. So she was actually the first person to meet me at the airport in Oregon when I interviewed for grad school.
Oh, yeah. I forgot about that.
She was doing a master's at the time with George Carroll in Oregon, and then she started her PhD in 2001 with me and a good cohort of people at the University of Oregon working in Karen Gilman's lab on one of the most frustrating bacteria ever invented, Helicobacter pylori. So Bethany got into looking at motility and quorum sensing and autoinducer production in Helicobacter and sensing in Helicobacter. From there, when we all diverged and went our separate ways after she was my roommate for
at least 2 years. I don't know.
So but I could. So it was so after University of Oregon, Bethany started a couple postdocs. The first one with Margaret McFall-Nye in Wisconsin, where again I have stories, but I'm not going to say. That is where Bethany started. Bethany started working with the squid vibrio system up there, continued with another postdoc in Spencer Nyholm's lab in Yukon, and then lastly went to Ann Dunn's lab in Oklahoma. So she's done 3 postdocs basically developing the squid vibrio system. This is the first squid vibrio talk I think we've had so far. So without further ado, Bethany Rader. Well, I will say everybody else on here in the Hangout, please mute your microphone, mute your camera. Without further ado, Bethany Rader.
Yay! Thanks, Dave. That was awesome. Yes, and I have things that I could get you with later on, so don't piss me off. Anyways, okay, I'm gonna try and get this going. Let me see if I can do this right. Can everybody see this?
You're good. You're good.
Yay! I got it to work. Hopefully it won't totally crash on us. So as Dave said, I am a new professor at Southern Illinois University, which is in Carbondale, Illinois. And I've been here for about 1.5 years, and we finally are getting the lab up and running and producing data, so that's really exciting. But today I'm going to tell you a story that most of this work has been either during my postdoc, well, one of the many postdocs, and then a little bit done here as well. And that's looking at alkaline phosphatase modulation of lipid A signaling in this model system. And this is going to seem like there's going to be a lot of background information, and I just really don't want to avoid that because I really love the system and I really want to point out the really cool parts. So there may be a little intro heavy, but hopefully it'll all work out. So what's great about this audience is I don't have to point out that we are mostly microbes. Unfortunately, I can't say that we're 10 times more microbe than human, but that doesn't take away from the fact that we live in community with millions of different organisms, and we are in fact a holobiont. So we are a collection of individual biologies that make up one biology. And contained in that holobiont is what's called a hologenome. So our genome is enhanced by, I think enhanced, by all the microbes, all the individuals that live within us. So we have this hologenome to go along with us. And what that really means is that our microbial communities or our microbiome can have a huge influence on our own biology. And we know this over the past 10, 20 years of work. When people first or actually really started focusing in on this, we've learned that microbes help regulate organismal development, regulate immune development, influence disease states, and some of the more recent work is showing that it's actually able to influence our behavior or our emotional states. It can signal directly to the brain, which is still something that completely blows my mind. And this is a very short list, and the list is ever expanding as we look more into this. But what I think this list illustrates is that how we obtain and then retain our beneficial microbial partners really matters. And so I think we not only should we continue cataloging what's there and understanding how they influence our biology, but we should also start looking at how we obtain our our microbes, and how do we convince them to stick around. Okay, and that's sort of the focus of the research in our lab. So sort of broadly, we're looking at how specificity of association is established and then maintained over the life of the host. And we're asking questions similar to how do the host's innate immune system and symbiont interact to establish a relationship. Of course, the immune system is classically what we think of as the body system that interacts with our microbes the most. We're asked how do host and symbiont communicate to each other to establish the relationship, but also to adapt to changing conditions. Excuse me. And we are, specifically what I'll be focusing on today is the question what host microbial or microbial factors enable this colonization and maintenance of symbiosis. And of course the answer is going to be many. There are many different factors, but for this talk I'm going to focus in on a microbial factor, lipopolysaccharide, and a host factor, alkaline phosphatase. So just a little background on alkaline phosphatase. I love these proteins because they've been around. I mean, obviously they've been around for a long time, but we have been looking at them for a very long time. So probably over 100 years we've been working on alkaline phosphatases, first identifying them, understanding their biochemistry, and then utilizing them for molecular biology purposes. So if you've done a lot of cloning, you've probably used an alkaline phosphatase. We also use them in the medical field. So if you've done blood chemistry or had blood chemistry done, one of the things they look at is alkaline phosphatase levels, and that can indicate a disease state. So they've been around for a very long time, and I love that about them. So they remove inorganic phosphates from a variety of substrates. So in our bodies, pretty much organic molecules, non-organic molecules, anything that needs a phosphate. And it does so in an alkaline environment, hence the name. They're ubiquitous across cellular life, so from bacteria to us, they're sort of always there. Though I haven't heard of a viral alkaline phosphatase, and if anybody knows one, they should send me that information. Ourselves, we have at least 3 of them. One is a tissue nonspecific variety. It's found in liver, bone, and kidney, and plays some role in bone mineralization as well as an indicator for hypo or hyperphosphatemia. There's a placental variety, which is expressed in placental tissues and is a marker for malignancy in different types of reproductive cancers, although its biological role in the placenta is relatively unclear. And then there's an intestinal variety as well. So this is expressed in the intestine, and it regulates the intestinal surface pH and helps in the absorption of lipids. But the function of this protein that I'm interested in is the fact that it can detoxify bacterial lipopolysaccharide. Okay, and what do I mean by detoxify? So in order to understand that, we have to take a brief moment and remember the LPS structure. Again, I'm preaching to the choir, but just bear with me. So if we take a gram-negative bacteria and we look at its membrane, it has an inner membrane, a peptidoglycan layer, then an outer membrane with a lipopolysaccharide. And if we look at LPS itself, it's made up of 3 different compartments, an O antigen, a core polysaccharide, and the part that's really important for this talk, lipid A. And I'm not going to go into about chemistry or this, any more of the structure of LPS than this. But I do want you to notice that attached to these 2 glucosamine residues are phosphate, ones at the 1 position and ones at the 4 position. And since our alkaline phosphatase strips phosphates, AP is able to remove these phosphates, and it's the removal of these phosphates that detoxifies the molecule. Okay, and so what do I mean by detoxifying the molecule? Well, it turns out that LPS, when it initiates an immune response, at least in vertebrates, is recognized by toll-like receptor 4, part of the innate immune system, and this binding to TLR4 or to its accessory protein MD2, which sometimes brings LPS to TLR4, is dependent on those phosphates. So if you remove those phosphates, you're removing the ability of LPS to interact with this innate immune receptor. So when LPS binds, TLR4 through MyD88 initiates a phosphorylation cascade, which culminates in NF-kappa B entering the nucleus and then turning on expression of a bunch of immune genes. Along with those immune genes, it actually turns on intestinal, at least in the intestine, alkaline phasphatase as well. So what you set up is this really interesting sort of loop where LPS is sensed, AP is produced, AP strips the LPS off, and LPS, TLR4 is no longer able to sense LPS, so you get less IAP. So so it's this sort of immune modulation. And so and so the activation or inactivation of TLR4 partially regulates immune homeostasis in the intestine. Okay, so that's great, but what does it mean in terms of, you know, us as functioning people? So we know from work in tissue culture that if you overexpress intestinal alkaline phosphatases in intestinal epithelial cells, you get less NF-kappa B translocated to the nucleus due to LPS. In vertebrates, administration of intestinal APs actually will reduce local or systemic inflammation and will improve tissue morphology, and this has been shown in a variety of inflammatory and infection models, mostly in mice, rats, and piglets. And in humans themselves, AP treatment has an anti-inflammatory effect on patients that have undergone surgeries and patients that have severe colitis and sepsis, so diseases that are at least in part associated with LPS. So why do I care about APs, right? So I've told you all this stuff about toxification and you know, inflammation and disease states in humans, but I study beneficial symbiotic interactions. So why do I care so much about APs? Well, if you think about your intestine or any other part of your body that's colonized by bacteria, some of those are going to be gram-negative, and you can imagine that there might be a large load of LPS present in that system that, if your immune system was reacting to, probably would cause some sort of chronic inflammation state all the time, so you'd lose that benefit. So APs to me seem like a mechanism for LPS tolerance due to that high load of LPS present in our indigenous microbes. And in fact, if we extrapolate out a little bit to the sort of nascent state, presence of APs may actually help shape the microbiota as those animals are initially being colonized by, again, modulating that immune response to that LPS load. And so these are the questions that we're asking actively in the lab right now. So sort of broadly, we're asking if APs have an effect on endogenous microbial populations. So will APs be present and functional in non-digestive organs that are colonized? And the reason I'm making the distinction of non-digestive is because I want to tease apart the sort of microbial tolerance function from the lipid absorption function or you know, pH regulation function. So so whether or not APs in the absence of those functions will still function to modulate symbiosis. And then one of the questions I wanted to know is whether or not APs are in a conserved mechanism of tolerance. So if you look, like I said, APs are sort of ubiquitous, so where they are, do they do this sort of mechanism? And then we're asking whether or not APs actually shape the microbiota by playing sort of a protective role during the colonization process. Okay, so to answer some of these questions, one of the things you need is the right type of model system. And so I always throw this slide up because I drew all those pictures and I'm very proud of them. But this is just sort of a snapshot of the different some of the different model systems that are used to study symbiosis. And one of the things you'll notice is that in most of these, it's actually a gut symbiosis that's being studied. So, so most of these aren't going to work for this type of study because remember, I want to pull it away from digestive organs. Of the two that are left here, Hydra is just a fascinating system, but they have a consortial microbiota, whereas I want to be able to ask very, very driven questions, very specific questions. So I wanted a system that's much simpler. And that leaves us our squid model, and it meets the criteria. So it's simple in association, has one host, one microbe. The microbe is a gram-negative symbiont, and it does not occur in an intestine. So to have you meet my model system, here is our host, the Hawaiian bobtailed squid. It's the Euprymna scalopes. It's endemic to the Hawaiian Islands, so it's hard to be me. I get to go to Hawaii to collect my animals several times a year. If you took this animal and flipped it ventral side up, which we did here, and opened the mantle, you would see this organ, which is an amalgam of symbiotic organ, ink sac, which you can see the fact that it's mostly black, and hindgut, which you can't see in this picture. But if you took this organ and you just sort of took off that top layer of ink sac, what you would see is this, and this is what's called the light organ, which is the what houses our bacterial symbiont. And I could talk at length about the cool structures in the light organ, but the one that really matters for right now for this talk is this sort of snot-looking tissue, which is called the central core. And inside the central core lives vibrio fischeri, which is a gram-negative marine bioluminescent bacteria. Okay, so those are the partners in this association. For just 1 second, I want to really promote my system because it's awesome. So it's a great model system for a variety of reasons. This squid is easy to maintain in the lab. So here's a picture of squid in a tank. This was in Spencer Nyholm's lab when I was a postdoc. But this symbiosis is horizontally acquired, so the animals will hatch into the seawater and will be without vibrio fischeri, and then the animal will have to recruit vibrio fischeri from that seawater in order to be colonized. And we can choose whether or not we want to colonize those animals. So the symbiosis itself is manipulable. And then even though a squid is not really that similar to a human, the symbiosis is similar to what goes on in our intestines. So here is a TEM of what the light organ epithelia and lumen look like. And what you see is you have an epithelia that has lots of microvilli, similar to a lot of our organ systems, and then the bacteria are packed in there, as my first postdoc advisor, Margaret McFall, and I would say, cheek to jowl in this lumen. So you have bacteria that are in tight association or in direct contact with the microvilli of an epithelial layer. So similar to what's going on in a lot of our body parts. And of course, vibrio fischeri itself is its own model system. It's extremely easy to culture in the lab. It's super easy. So sorry for all of you that work on things that are hard to grow. We can just put it on a plate, stick it right on our bench top, and the next day we have fabulous growth. It's genetically manipulable, so and over the last maybe 30 years, it's been a model system. We now have a library of different mutants that we can work with, and we have tagged them with all sorts of fluorescent molecules, and so we just have a treasure trove of strains to work with. And then thanks to Mark Mendel at Northwestern and other researchers in the system, we now have several actual genomes of symbionts from squid. So we work with a type strain, ES114, that's what we colonize with in the lab, but we also have in isolation and sequenced other isolates from the squid itself. So fantastic system. Okay, so the first question that people always ask is, why do squid have this organ that's colonized by one type of bacteria? And the answer is in its day-night cycle. So during the day, our squid are quiescent, so they're nocturnal hunters. So during the day, they sort of bury themselves in the sand and chill out. And of course, they're really good at it. So you wouldn't ever see one on the seafloor if you're walking through the shallows, and their predators can't see them either. But since they're nocturnal hunters, they come out of the sand at night and forage for food. And even though it's nighttime, there's enough ambient star and moonlight that sort of filters through the water column that the animal actually casts a shadow And it's not a shadow that our eyes would pick up, but its predators have eyes that are attuned to these things, and if a shadow is cast, they will sense it and eat the squid. But if you remember that light organ is ventrally facing, so the bacteria inside are all quorum sensing, they're all producing light, and that is being focused downward, not only because of the position of the light organ, but the animal can actually modulate the light that's being emitted from that light organ so that it completely matches that ambient light coming from above, and therefore hiding their presence from predators. And this is a process called counterillumination. And it's such a tight association that we've never found a squid in the ocean that didn't have the vibrio fischeri, bioluminescent vibrio fischeri in its light organ. Okay. So that is one day-night cycle. There is a second day-night cycle, and that's important to illustrate for sort of the work that we do, and that is a sort of symbiont population day-night cycle. So during the night, when the animal is out foraging for food, the bacterial population has to be pretty dense because light is a production of quorum sensing, so population density has to be high. Oops, sorry. Population density has to be high. And at dawn, there's actually a light signal that the animal perceives through its eye that communicates to the light organ, and then the light organ will squeeze out about 95% of the lumen of the organ itself, and so about 95% of the bacteria. And you can sort of see that in this TEM of the light organ just post to that, what we call venting behavior. When the animal's quiescent during the day, that population will increase again, such that at nighttime, when it comes back out to forage, the population density is high and they're luminescent. And so this is sort of like the light organ is sort of like a living chemostat, keeping that population going. Okay. One other thing I want to explain before we go on to the actual sciencey part. So the other sort of hallmark of this association, and what's been studied really intensely for the last probably 20 years, is what happens during colonization of this organ. So here's a picture of a juvenile squid. They're super adorable. They're basically little tiny adults, almost fully developed except for the light organ. And if you took a look at the light organ from a juvenile animal, it looks different from what I just showed you for an adult animal. So the juvenile animal has an extra epithelial layer sort of on top of both lobes of the light organ, and it's ciliated. And I'm not going to go into detail about what it does, or all the biology that we know about it, but basically these structures are there to help promote colonization. And what you'll notice is that sort of at the base of one of these ciliated appendages are 3 pores, and that's on either side. These pores, if we take a look in the internal parts of this organ, lead down ducts and into what we call the deep crypts, which are the living spaces of the bacteria. So the deep crypt here coordinates with the central core that you saw in the adult. But obviously this is not the structure that ends up being an adult light organ. And what happens is once the animal reaches the internal part, or sorry, the bacteria, not the animal, reaches the internal part of the deep crypts, it sends a signal to the light organ to start undergoing an apoptotic program. And so you get regression of these ciliated epithelial fields again on both sides of the light organ. So here's a micrograph SEM light organs 1, 2, 3, and 4 days post-colonization. So these are symbiotic or sim animals, and you can see the complete regression of these structures, although at this point you still have 3 pores. Okay, and due to a lot of work that was done prior to my entry into this system, we know that there are at least 2 signals that trigger this apoptotic event. One is peptidoglycan, specifically the TCT monomer, and that's a whole other story that is also super cool, but I'm not going to talk about it. And the other is LPS. So now we've circled back around to this concept of LPS, and so it turns out that LPS regulates the initial stages of apoptosis. So Here's an example of a light organ that is aposymbiotic, so it hasn't been given either bacteria or any sort of molecule. If you give this animal bacteria, within 14 hours you get these pycnotic nuclei. So the nuclei are starting to condense, which is that early stage of apoptosis. And this is easy to visualize. You just throw some acridine orange or some other aethylene bromide derivative onto this, and you see that diffuse staining turns into this pycnotic staining, condensed staining. And you can see the exact same thing happens when you treat a aposymbiotic light organ with LPS. So LPS is initiating this early stage of apoptosis. So when I started thinking about alkaline phosphatases, I thought, well, this is a great system now to take advantage of, because we have this wonderful readout of whether LPS is being, you know, effective or toxic or not. Okay. So this was a really great place for me to ask, do APs actually play a role in colonization by beneficial bacteria? So finally, we're getting into the science. So the very first question I asked when I started working on the squid are whether there are APs in squid tissues. So at this point, we don't have a genome, although we're working very hard on that. So I went through and I mined the transcriptomes that we had available to us at the time. And I found two different APs, or two different isoforms. And I named them EsAP1 and 2. Creatively. They're about 90% identical to each other. They only differ at the very 3' end. I think it's like 25 amino acids are different in the C-terminus. And so we think it's a result, these isoforms are a result of splicing, but again, without the genomic data, we're not entirely sure. They're 53% identical to vertebrate APs, so not that close, but and I'll show you in the next slide, they're structurally similar to vertebrate APs, so we're pretty sure they're APs. And then I just did a quick look at where these things were expressed in an adult animal and in a juvenile light organ. And what we found was that their expression was different from one another. So AP1 seems to be expressed in tissues that we know are colonized by bacteria, whereas AP2 seems to be expressed in other types of tissue other than the colonized organs, except for in the juvenile state in which the EsAP2 also seems to be expressed in the light organ. But overall, EsAP1 seems to be more specific to symbiotic tissues, which is also really interesting because no one really does this kind of work in invertebrates. So as far as I know, this is one of the first descriptions of differential expression of APs in invertebrates. Okay, so here's the annoying alignment slide that I feel like everyone has to have in a talk at some point. But don't worry, I don't want you to look at anything that specific. This is basically showing you that squid APs have all the right things in all the right places. So here's the active site. It's conserved between squid, oyster, cow, and human. And then the arrows are metal binding sites, which are also required for activity. So squid APs have the right things in the right places. Pretty sure they're APs. But of course, we had to test that. So at the time when I did all these experiments, we hadn't cloned these APs for expression yet. So we couldn't ask specific questions about those specific proteins, but we could ask whether or not we see AP activity in light organ tissues. So what we did was we took central cores from adult light organs, we mashed them up, and we extracted the soluble fraction, so total soluble proteins from those light organs, and then we put them into your classic alkaline phosphatase assay that anybody that's taken chemistry has probably done at some point. We added a substrate called PNPP, and if there's an AP there, it will cleave off one of the phosphates and produce a yellow substrate, which then you can measure. So the yellowness of the solution dictates how much activity you have in your sample. And you can compare that to a sort of a standard curve of your PNP and get some more quantitative ideas about how much hydrolysis is happening to your substrate. So we did that with our central core soluble proteins, and we did indeed find activity that peaked at around a pH of 8 and then remained relatively high through pH 11, and was very low until about pH 6. So this told us that there was phosphatase activity occurring and that it was in the alkaline spectrum. So confirmation that we have APs in these tissues. Okay, so yay, we found that we had AP activity. But since we're trying to correlate this with the colonization process, one of the things we wanted to do was figure out whether or not these APs are actually regulated by the symbiosis. Because one thing that I showed you already is that LPS seems it needs to be, quote unquote toxic, at least in the early stages of the symbiosis, to induce that apoptotic state. And so we did the exact same experiment, but with soluble protein from juvenile light organs. And so on this graph, it's a little jumbled, but on top here is day-night. So black is night, open or white spots are day. And then we did this experiment using light organs from both symbiotic animals and non-symbiotic animals, which you might hear me also call aposymbiotic. I sort of go back and forth between those terms. And what we found was that if you looked at the non-symbiotic light organs, you didn't see that much regulation of AP activity over the first 72 hours of the association. And then, however, if you looked at sim light organs, in the first 12 hours, you didn't see very much regulation. There's a significant difference here at 6 hours, but the major regulation in activity that you see is starting at 24 hours, and then at 24, 48, and 72, you have this bump in AP activity. And if you take a look at this light day-night bar on the top, what you see is that this is occurring when the light organ, sort of at the transition from day to night, when the light organ is going to have a full complement of bacteria, so there'll be a lot of LPS around. And then conversely, at 36 and 60 hours, which is basically, I did this experiment right after the animal vented, there's not going to be as much LPS, and therefore AP, there's not going to be very much AP around as well. And then here's just a the inhibitor control showing you that the activity we're seeing is due to phosphatases. Okay. We wanted to, so that was awesome getting that result. However, we wanted to make sure this is also something that happens in adult animals. We want to see if these APs are regulated by the symbiont. And so we did a similar experiment, but since we have far fewer adult animals than we do juveniles, instead of doing over a series of days, we did several animals over one day-night cycle. And so again, day-night is up here, and we saw a similar but slightly shifted result from the juveniles, so here we see that there is a peak activity right before, this is about 2.5 hours before lights out, and that, but the light organ is going to have a full complement of bacteria, so there's going to be a large load of LPS around. Okay, so it seems that EsAPs are active in the light organ, and they are regulated by the symbiont in both juvenile and adult animals, which was great, except for the next thing we wanted to do was show that this activity actually correlates with where the bacteria actually are. So if you'll notice, here's our little map of our juvenile light organ. You'll notice that there's all sorts of different tissues here, and what I told you earlier was that the light organ is attached to the ink sac and to the hindgut. So it could be that when we just mash up whole light organs, especially the juveniles, where the AP activity could come from some other part of that tissue. So we wanted to correlate AP activity with where the LPS is going to be. So we used a substrate that's similar to PNPP, except for when the phosphate is cleaved, it produces a fluorescent substrate, or fluorescent precipitate, basically, that we could look at. And because we have such a wealth of bacterial mutants and strains to work with, we have an RFP tagged bacteria. So we could actually look at AP activity via fluorescence and coordinate that with presence of the bacteria. And so when we looked at a 48-hour light organ, a non-symbiotic light organ had some activity AP activity. That was mostly on the surface on some of these ciliated epithelial cells, and a little bit in these sort of part of these duct structures, sort of leading into the duct. And this is something that I'm actually really interested in and hope to go back and look at. But you know, there are no bacteria, and it doesn't look like these have actually gotten into the duct, or the deep crypt spaces of the light organ. If we look at a 48-hour sim animal, we see that there's a huge increase in activity, which is great because it correlates to what we saw in the juvenile activity assays and the adult activity assays as well. But what you see is you start to see, so you see enhanced fluorescence where we saw it previously, but you start to see now fluorescence in the crypt, or the deep crypt spaces. And if we look at where the bacteria are hanging out in this light organ, we see it's basically in the same places. So the fluorescence or AP activity actually correlates to where the bacteria are. Okay, so now we know that AP is there, they're active, they're regulated by the symbiont, and they are in the places that the symbiont is going to be, or where the LPS will be. So the next question we had was whether APs could actually affect vibrio fischeri LPS. So could we, quote unquote, detoxify vibrio fischeri LPS if we gave it AP? And since we didn't have our EsAPs cloned and expressed yet, we went to our calf intestinal alkaline phosphatase, which is what we used in our alignment. So we know that that our APs should act similarly to this calf intestinal AP. Okay. So we did a really simple experiment where we took juvenile light organs and we gave them either the bacteria, lipid A from vibrio fischeri, or vibrio fischeri lipid A that had been incubated with calf intestinal alkaline phosphatase. And what we found was really exciting. So if you look at our non-sim light organ, you see, again, using acridine orange, you see that sort of diffuse staining. With your sim, you see punctate staining, and this light organ was super sim because in 18 hours this epithelia has regressed quite quickly. If we gave an animal, a non-sim animal, just regular fischeri lipid A, we see that same entry into early stage apoptosis. But if we treated it with CIAP ahead of time, it looks like a non-sim. So we got rid of that ability of the cells to enter into apoptosis. So it seems like APs can affect lipid A signaling. And so here, this is just the quantification of counting all those tiny spots. Okay, so now we know it's there. We know AP is there. We know it's regulated. We know it can affect B. fischeri lipid A. The next question was whether or not APs actually are required for some part of this process, either the colonization process or persistence of symbiont in this organ. And it turns out that it does. And so we, along with not having a genome, sort of the other downside to the symbiosis is we don't have any ways to do reverse genetics yet, though I think CRISPR is going to be what's going to get us there. But that's an aside. So we can't knock out APs in the squid, but what we can do is inhibit them with levamisole, which is an AP inhibitor. It's used as a medicine to treat all sorts of things that, well, we'll talk about later. But so we can specifically inhibit APs, and so we did. So we took juvenile animals and we gave them the bacteria as well as the inhibitor and looked at how many bacteria remained in the light organ over the next 2 days. And so we can look at bacterial content of the light organ in 2 ways. One is we can just plate them, freeze them and plate them to give us a colony forming unit count. The other way we can do this is by looking at bioluminescence. So Again, it's not something our eyes could see, so unfortunately, if you have a squid in a dark room, you won't actually see the bacteria glowing, but you can pick that up with a luminometer. And so we did both of those things, and what we saw was that by 24 hours, the treated and the untreated and the inhibitor-treated squid were colonized by bacteria. The inhibitor-treated, though, to a lesser extent, and that by 48 hours, the animal had basically started kicking out the symbiont. So close to little to no bacteria left in those light organs. And if we took the light organs from these exact animals and plated them, and we did, in fact, see this dramatic decrease in CFUs over CFUs in the light organ. And just to convince you that this was an effect of the inhibitor, we cultured vibrio fischeri in the presence of the inhibitor and saw no difference in growth. So it really seems that regulation of AP activity is very important to the symbiosis. So it is required for persistence of the symbiont. And that was really exciting when we got this data, and then we started thinking about, well, how does this actually work? So what is the mechanism that regulates AP activity so that persistence is maintained in this system? Because you don't want to have your AP activity off all the time, which is what this is showing, right? But you might not want it on all the time either. And so part of the answer to this, we think, comes from a paper, a really great study that was done, oh man, 6 years ago now, from a microarray that Andrew Weir performed. He was a postdoc in Margaret McFalney's lab, where he did this really fascinating study where he did a microarray of both the host and the bacterial tissue at the same time, so he could sort of see what kind of coordination was going on. And so everyone should go read that paper. But what's most important for what I'm talking about is that if you look at genes that are involved in metabolism for the bacteria over this day-night cycle. So here's, and this is an adult animal, excuse me, I should have mentioned that. So here's day-night cycle again. What you see is that at some point during the night, the bacteria switch from aerobic respiration to chitin fermentation. So there's a shift in their metabolism to fermenting chitin. And what that does, at least in theory, is start acidifying their environment. So a lot of the byproducts of chitin fermentation are acidic. So, and this has been corroborated with work done in the lab from Ned Ruby's lab, where if you take the bacteria in culture and force them to ferment chitin, they acidify the culture medium to about a pH of about 5.5. And there are a couple other lines of investigation that have corroborated this idea. And so this actually sets up a really amazing working model for us, which I'm going to go through hopefully pretty quickly here. So here's here's one day-night cycle in this sort of silly graph. So we have bacterial density over the day-night cycle. We've seen, I showed you that already. If we add in the data that I just showed you from this microarray study, and we look at the pH of the light organ, what we should see is the pH during most of the day and the sort of first hours of night will be relatively high, and that will drop as the bacteria switch their metabolism. And if we, on top of that graph, add the AP activity that we saw in adult light organs, what we see is, again, high activity at the end of the day, early night, which then drops somewhat coordinately with this drop in pH and this change in bacterial metabolism. So what we think is happening is in the morning, everything's expelled from the light organ. Both the pH and AP activity slowly build. They get to a peak. Bacterial change metabolism. The lumen is acidified. APs no longer work because of the acidic environment. And then that causes LPS to then be more toxic, right? Because you don't have dephosphorylation of that toxic. And at that point, the LPS is able to initiate an immune response, which we think is, at least in part, responsible for the venting event at the next dawn. So this is actually a great model, and we're really excited because we just are about to receive funding to actually test this model. So we're really excited about that. But what's really nice about this story is that it really works out, right? So during what we found in the juvenile light organs is that during colonization, lipid A needs to act as a signal, so we didn't see very much AP activity before 24 hours. And then once that rhythm, this sort of dial rhythm of the light organ is established, AP activity can then allow for tolerance of high levels of LPS. And then once the pH drops, signal for effacement and venting. Okay, so if we go back to the original questions I was asking, way back, to the beginning of the talk, we were asking the questions, do APs have an effect on microbial populations? Specifically, will APs be present and functional in these non-digestive organs? And the answer was yes. APs, in fact, are required, at least in this system, for the persistence of these bacterial communities, and they are regulated by the symbiont. And we asked, are APs a conserved mechanism for LPS tolerance? And my answer would be yes. APs probably have a similar function in vertebrates and in invertebrates and vertebrates, too many ins. So it does seem, at least from this very small sampling, to be a conserved mechanism for LPS tolerance. Okay, and then the last question was, do APs shape the microbiota by playing a protective role during colonization? And it seems, at least for this system, not so much, right? Because, like I said, LPS needs to have those phosphates in the first 12 hours of the association. So probably not playing that much of a protective role in this. But we can expand this question to ask, what about humans? And so hopefully, I'm going to go through this really quick. I know I'm sort of going long on time. But we've sort of expanded our thinking about APs and their role in colonization to humans. And so one of the questions we want to ask is whether APs help protect the infant intestine from aberrant inflammation during colonization. And our sort of model for aberrant inflammation is necrotizing enterocolitis. It's a disease characterized by intestinal inflammation. It primarily affects premature infants and can lead to bacteremia, endotoxemia, and mortality. So it's actually a really, really bad thing. And in mouse models has been associated with colonization of the intestine. It's correlated with TLR4 signaling, so with LPS signaling. If you actually treat it with intestinal alkaline phasphatase, that will decrease inflammation. And this is in mice models that are derived to be NEC models. So one of the questions we wanted to ask is whether or not the amount of AP in the infant intestine is correlated with the development of NEC. And our first step was really to characterize the AP levels in the infant intestine because no one had actually taken a look. And so this is work that's done in collaboration with Karen Gilliman at U of O, Jennifer Smilowitz at UC Davis, and Ye Yang at University of Florida. And so when we were thinking about it, there's actually two possible sources of APs in the infant intestine. One is infant-produced intestinal alkaline phosphatase, and the other is AP that's produced and supplemented in mother's milk. And we know that mother's milk has AP because it's one of those things that is checked for after donor milk has gone through pasteurization. And so we hypothesized that the infant intestine will probably increase in AP amount as the intestine matures, whereas mother's milk will start off high in AP to supplement that intestine and show a decreasing trend over time. And what's excitingly, that's exactly what we found. So work that Ye Yang had done looking at meconium from infants of different gestational ages showed that preterm infants had significantly less AP in their intestine than term infants, probably due to the immaturity of the intestine and the immune response in the intestine. And we found that we saw that, the opposite correlation with APs in mother's milk. So we had about 80 different mothers that gave samples in week 1 to 3 to 4 and 6 to 7. We found that AP in the first week was significantly higher than in any other week. And if you look at just the days within that first week, the closer to birth, the more AP was actually in the milk. So colostrum had obviously the highest amount of AP. And what was really exciting about these data is that every single mother had the exact same trend, even though the absolute values of AP in the milk were different. There was always more milk in the first, or AP in the milk in the first week than in subsequent weeks. Okay, so for this short little thing that I showed you, we're in the very beginning stages of this. We're trying to publish this first data, and hopefully we'll be able to expand this study. But we're concluding that AP activity in the intestine of term infants is high, sort of during the normal time frame of when the intestines are going to be colonized, and that the premature infant is probably at more risk for inflammation due to colonization because of the lack of either intestinal alkaline phasphatase from their own bodies or AP from mother's milk. And that's because a lot of premature infants are fed formula, which is not supplemented with AP, donor milk, which is pasteurized, so there's no AP activity, or fed intravenously. And so we have a lot of future work that we want to do on this. Of course, ultimately we want to assay for AP activity in preterm babies that have been affected by NEC. Okay, and with the end of my talk, I just sort of want to think a little bit more broadly and leave you with food for thought. And this might be me just waving my hands in a crazy manner, but this work on APs has really brought some weird questions to my mind. Like, for example, did symbiosis shape the evolution of AP function? Which seems like a crazy question because we know what APs do, right? They take phosphates off things, and of course that's important for a lot of different things that happen in our own biochemistry. But the fact that we evolved with our microbes, so the minute there was a multicellular organism, there were probably microbes associated with it. So there is this huge possibility that AP function might have actually been shaped in part by those symbioses that are, have been a part of us all for this evolutionary ride through time. And then of course, the bigger question is, are microbes a potent selective force? So we think a lot about whether or not hosts are selective forces on microbes, and there are a lot of people in our system and other system using the host as an incubator to see how bacteria will evolve over time, right? So these sort of artificial evolutionary experiments using a host as a selective force. But is the opposite also true? Are our microbes a potent selective force for how our biology works today? And then something that I think we need to start doing is to start thinking about human biology in terms of evolution in the context of symbiosis, right? So, not only not only just how our immune system functions or things that we've already thought about, but almost every part of our biology. Okay, with that, I need to thank my lab, my sources of funding, the NIH, the American Heart Association, which let me work on squid as a postdoc, and then the small amount of money I've gotten from SIU, and then of course my collaborators on both the infant project, and then of course Margaret and Ned on my squid projects. Here are the references in case you're interested, and with that I will take questions. Oh shish.
That's awesome.
Oh good.
Yeah. That
okay.
was I've actually never seen a whole squid lecture before, so that was I now understand it a lot better than I did before.
That's awesome.
Yeah, no, I lectured on quorum sensing and like LuxR and LuxI today too, so
that is, did it go well?
It was fine. Like, yeah. Couple questions though. So, you have chitin, the mutants that can't ferment chitin, and what do they do in like venting?
So they, so if you make a mutant that doesn't ferment chitin, they will persist in the light organ for the first, as far as I understand it, like 4 weeks of the association, and then after that 4 weeks they get kicked out. So 4 weeks is a really interesting time point in the life of a squid, because that's when it goes from not responding to daylight in terms of just their behavior to bearing during the night, or during the day. So something weird is going on at that time frame. But so these mutants will colonize, they will vent as well, but they will eventually be kicked out by the bacteria, or by the host.
And then one other question. So I know like there's other vibrios that colonize light organs and other things that do they, so like in the fish, do they vent every day, or are they different in terms of like metabolism and stuff because of that?
Yeah, as far as I know, this is the only symbiosis that undergoes this daily venting. But don't quote me on that. I'm not in, I have not, I've not looked at all of them. But yeah, I think that for other organ systems that have fischeri or other types of luminescent symbiotes, I think that they don't. So one of the things I didn't mention is that there are these blood cells in the squid that supplement the light organ with chitin during the nighttime. So I don't think other systems do that. I think they're just always producing, giving them glucose and other sugars that they can use for normal metabolism. Oh, I lost you. There you are.
I'm still here. I'm looking online. Does anybody else have questions in the Hangout here, or no?
No.
Nothing online. So yeah, no, that was awesome though. I am. Good job.
Thanks. Do you like APs as much as I do now?
I'm jealous. I'm jealous.
We should collaborate.
Good.
Yeah, it's really fun. We have a nice system up and going here. So yeah, anybody that wants to collaborate on squid stuff, let me know.
Cool. Well, I'm gonna sign off and then we'll chat afterwards. But
okay.